There we go. So viewers, apologies for the sunshine streaming through the window. I promise it will move in seconds. There's trees outside. Cool. So Daniele Marinazzo, finally. So yeah, man, this is great. I've been trying to sort of maintain a sort of a theme. And obviously confounds is what I've been preoccupied with for a while, probably a decade. And it's just been something that seems to have not really moved as much as I would have hoped in the field. It still seems to be more of a sort of a marginal peripheral endeavor. And, so many people now doing things like, psychedelics and other things. And it's just like confounds, just like, coming out of the woodwork. So, I've been really fascinated by your approach. I know that you are in the church for corrections and you're doing, LFO corrections with rapid tide and stuff. So take it away. What's your perspective right now on this and how it affects what you do?
Yeah, so say that from my point of view, so in general, the approach to neuroscience has started with, let's say, after my PhD in physics, but in sort of physics which had nothing to do, let's say, with the MRI physics. So it was more statistical physics. So it's more about signal processing, but also collective behavior. So systems made of multiple units, and we record from those different units, and then from the global recorded dynamic, we try to make sense or so. So let's say in neuroscience, basically what has been called brain connectivity, if you want, and then the brain functional connectivity when you put the dynamics into it. So let's say that this is how I entered the field. Then there were some moments in which I was more into electrophysiological data, other times more into fMRI or so. This also speaks to the fact that indeed it's more an approach based on addressing some issues in the dynamics rather than on a specific modality.
Okay.
This also incidentally makes me feel somehow as an imposter in both fields. So definitely in this case, and especially looking at the previous lineup of your guests, definitely the imposter syndrome is pretty big for me in this case. Still, I'm let's say naively confident that a sort of maybe external or different type of view can be helpful in some cases.
Yeah.
So for me, let's say one of the approaches I started working with in brain connectivity and in brain functional connectivity was what was previously and now still sometimes called as effective connectivity. Actually, a sort of a misnomer in the sense that, let's say, there was a period in which functional connectivity was the thing that you do with correlation, and then effective connectivity is when you put a directionality in it. Some people started calling it this way, and maybe I also did it in the past. On the other hand, let's say effective connectivity reflected the terminology started first in cybernetics by Ad Artsen, and then used also by Carl Friston and others when they did some generative models of brain activity. So in this case, the effective was, let's say, when you have a generative model. But since, of course, a generative model, if they want to be closer to reality, and then the reality is basically directional, even if
Oh, I think I might have lost you.
It was collapsed the name effective connectivity with the directional. Then on the other hand, there is actually no much difference in the approach of functional connectivity in terms of statistical dependencies, whether they are directed or not. So anyway, when I started, I had PhD to improve the so-called Granger causality, which is basically a measure of statistical dependencies based
Oh, interruption. We'll hopefully recover.
connectivity issues in neuroscience, and then of course it got a lot of pushback for some good reasons. But on the other hand, there was a moment where I was puzzled that all the pushback that came into Granger causality was not used, for example, for correlation. So a lot of people were happily using correlation and Granger causality, which, okay, you cannot call it causality if you want, but then we come back to the issue of causality. But at least you are correcting from on the previous history of the target time series, etc. So it's arguably better than the correlation, but that was the one, let's say, suffering for most pushback. And one of the arguments for this pushback, when applied to fMRI, was the hemodynamic response. So there was a famous paper by Olivier David and Carl Friston and the co-authors, where they used some rodent data in which then fMRI data. So no longer you had an hemodynamic response, but you also had, let's say, quite long hemodynamic response and so actually they showed that indeed the fact that the neuronal response is filtered by the hemodynamic response function and is also delayed, and also the hemodynamic response function is different in height, in length, in different brain areas, then this would confound the true neural connections. Now, also for true connections, then we will maybe open another parenthesis later. So just to
that's leaving out all of the physiologic confounds. That's just saying just the neurovascular coupling is sufficiently variable across the brain and is sufficiently lagged that you can't make meaningful estimates.
Yes, exactly. So let's say if you use an approach based on temporal precedence, which is what Granger causality does, then if you don't take this into account, then the thing will be confounded. Then we will. There are some nuances to this, but for the moment, let's go to the. Let's just stick to this first uncomfortable truth. Then, one. To address this main criticism for Granger causality applied to fMRI. Then, and so our approach was based on point process theory and based on some previous work, mainly by the group of Dante Chialvo, who had worked on let's say criticality before, and then he had seen these peaks in the bold signal as some signature of some neuronal avalanches at the neuronal level. Then let's say the results still hold, but the fact that these peaks are generated by neural avalanches has been somehow reconsidered because let's say it's a general properties, let's say, of co-varying time series that let's say the extremes are more correlated than the rest of the points in the distribution. So let's say this same approach, which then led to approaches such as the co-activation patterns by the group of Jeff Down and Xiao Liu. So let's say basically spatial correlation, spatial clustering of bold peaks, which recapitulated the resting state networks, if you want, let's say, so this is, let's say, the general framework under which we developed our approach. So the idea was that every time that you have a peak in the hemodynamic range, so let's say in the same range for which you that you use when you filtered prior to applying a rapid tide, so let's say up to 0.01 to 0.08, 1 hertz, etc. So whenever you have a peak there, we can suppose that it is due to some pseudo neural event, which then triggered a hemodynamic response. And so if you solve a general linear model, when you try to fit all the peri-peak intervals, so you take some time, some points before, some points after each peak, you take these segments and you fit a general linear model when you have some parameters such as the delay between the neuronal pseudo event and the peak and the shape of the response function. We will come to this later. Then you can have an estimate both of the lag between the peak and the event, so whenever an event would have happened, and then the shape of the HRF. This, of course, is.
Let me just ask for a point of clarification, because I don't know causality or modeling, but first of all, you're assuming that you can point an arrow, that it's meaningful to have a direction. So I can imagine that's the case for something bottom up, like your visual cortex. You've got retina, occipital lobe, LGN, awareness, etc. Right? Then processing, right, and then it can be higher functionality. So there, at least the bottom part of the plumbing clearly has to go bottom up. But for many processes in the brain, and maybe resting state is one, isn't it always re-entrant? I mean, how do you even break into this as a cycle? Isn't this like looking at traffic between multiple cities and trying to determine whether it's rush hour?
Yeah, no, completely. So indeed, this approach I was just describing is a univariate approach. It's not about connectivity. This is about every single voxel. So what happened then? Indeed, we try to address the connectivity approach first with the convolved time series, but also between the neural events. So to see whether, let's say, a neural pseudo event detected with our method, for example, in the insula, if you do an histogram of the relative timing between a peak in one region, region A, and a peak in region B, you can say whether, let's say, the peak are unrelated or one tends to happen after the other.
So, on the average, so you're not to try and do some brain-wide thing. For these two areas, A follows B or B follows A on, given all the other limitations.
But let's say, even if, let's say, the primary reason to develop our approach was to, let's say, have a better, a less confounded Granger causality estimate by deconvolving the HRF, then lately we turned out to use it more locally, so to have a shape of the HRF which could inform about several things. But the idea was that one, so to get rid of this temporal confound, and then with these pseudo events, then you can also do something based on as if they were spikes, let's say, as if they were.
And you can check it using some, like you could do, does LGN precede higher cortical, etc. Right, because otherwise it doesn't make sense.
Now this we did it I guess only once on a paper on chronic back pain or so, where we try to have influence between two series using the peaks, and this would definitely be something that is interesting to do after correcting for blood arrival time and all this. So since it's been a while, we haven't done it anymore, but this is something which I really would expect some difference. We will come back to this when we will discuss, let's say, what happens usually to my data before and after the application of R60. So the idea of our approach was this one. And is an approximate approach. It has some limitations. For example, I mean, one clear limitation is that it assumes, let's say, being based on point process, it assumes that there is a neuronal event triggering hemodynamic response. So, for example, you cannot apply it to breath hold data, because there you don't have, let's say, the assumption that a neuronal event will trigger an hemodynamic response, because you don't have a constant oxygen.
No, but you could ask the equivalent question, which would be, are you later or earlier in the vascular tree, right? You could use it as like a vascular mapping method, right? If ignore neurons, like Blaise says, the neurons just hold the blood vessels up. So, but you could you not, because it's regional, right? If you look at MCA, there's quite clearly a nice flow there, and the later area has to the downstream area has to arrive later than the earlier area, right? Or the plumbing doesn't make sense. So why does it presumably it's not essential that there's neurons in here, it's information flow, right?
Yeah.
So if you haven't corrected for this and you're looking for neurovascular coupling, you've got to confound, because the chances are the variance in the blood gases is much bigger, in the low frequency oscillation is much bigger. That's going to drive everything. And then if you now change this and you say, okay, forget the neurons, I'm going to do a breath hold. I want to know, that's an event still, right? So why wouldn't your method still work for that?
Yeah, I mean, let's say the one assumption would still hold. Let's say the assumption of a transfer function. So you have a transfer function between. So if you think in terms of state space models, in which you have the unobserved neural model and then the observed level, which is the bold signal. So then that transfer function, which is the neurovascular coupling, still holds. Then the fact that let's say, I mean, the confidence in the fact that some neural activity, so let's say, basic fMRI 1.0 would recruit, let's say, would make a region more metabolically active, and then the need for glucose would also increase the hemoglobin, etc. And so, let's say, this becomes a bit more blurred, let's say, if you want. Then there is no, let's say, black or white, but let's say all the things like being downstream with respect to the regular blood flow, etc. Or the standard models of the HRF could maybe change, because then one other thing that I just briefly mentioned before is that in the general linear model that we use, then you need to use some parameters from the shape if you want. So either you can use the canonical shape with the derivatives, or you can use a set of basis function like a mixture of gammas, etc. Or you can be most as agnostic and then use a finite impulse response. And then in the sense you just fit whatever the response is without, I mean, without constraining with more parameters.
And I assume that you are assuming a stationary hemodynamic response that you're trying to fit out of your 5, 10, or 15 minutes data.
Yeah, exactly. So yeah, let's say that the other limitation actually is a twofold limitation. One is that indeed, in this case, you assume that there is stationarity over the length of the scan. Then, of course, if you have a very long scan, then you can split it, etc. or so. But the idea is that in order to reliably solve a general linear model, then you need, I don't know, a dozen of these peri-peak intervals, let's say. And so you take over. And then the second one is, of course, if what would happen if you have two fast neural activation within the same repetition time. Then, of course, in our model, this would collapse into one, of course, because it works at the TR resolution, let's say.
Yeah. So there's an intrinsic temporal smoothing right there.
Yeah.
Okay.
I mean, and this, I mean, on one hand, it should not be a surprise because ultimately it's a data-driven thing, so you cannot create information from nothing. So let's say if you have the bold time resolution, yeah, it's difficult. You can extract faster things there, but then maybe with the Diana things will improve.
Okay, all right. Okay,
So,
I'm starting to get a much better sense now of what you're trying to do. Yeah, cool. All right. Well, yeah, anything else that
yeah, so I mean, this is as it concerned then the resting state HRF thing. So then after this, so in the first paper in 2013, we used it for Granger causality. Then there were some follow-up papers, some by our group and some together with the group of Gopi Deshpande and Ranga Prakash in the US, who did some work there, also on clinical population, on rodent population, etc. And in particular, one in which they showed quite clearly that is not only for lagged influences such as Granger causality, but also correlational approaches are confounded by the shape of the HRF.
right, yeah, okay, which is stuff that Dan Hamworker and others had also
Yeah,
looked
exactly.
at in aging.
And that's it. But then we started using it more, let's say, as another feature of bold activities, such as, let's say, I don't know, the low frequency fluctuations, the variance of the signal, so some local measure which is modulated in some conditions, such as, I don't know, loss of consciousness, arousal, other things. And then ultimately,
also on the assumption that the
yeah.
other the physiology, the systemic stuff is not driving all of this, right? Because
yeah, I mean, imagine that you have a bold data set and then you observe with a simple first level, second level analysis, some differences in I don't know, in some regions of the brain. Then the question can be, okay, are those neuronal differences or are these hemodynamic differences?
and
Then
hemodynamic,
yeah, of course,
it's an agnostic term.
yeah, of course, and of course the two things are so intertwined that you cannot give an answer, because then even if you estimate HRF and then you deconvolve it, you don't have the neuronal activity, because still, if you are deconvolving it, I mean, even when
you got an inverse
you're
problem
removing
right
stuff, you are actually inducing some shape, so you cannot really have the two things separate. But let's say the idea is that whether there is this, let's say, static, if you want, measure of this transfer function between the two states, the unobservable and the observable one, which is modulated by a specific task, rather than the whole dynamics or both things at once. But of course, the two things are not separate.
Okay, so you said task. Does this work on task fMRI? And if not, why not?
So in principle, it works on task fMRI. In our case, let's say the key part of the algorithm is the convolution. So you estimate basically the ones in the design matrix. Then, if you already have the design matrix, then you can just estimate the HRF with the basis function as you already do. Then, or you can do it something mixing up the two things. So, for example, there is now an approach being developed for event-related potential in EEG or MEG, where also, even if the dynamics is faster, you can still have, for example, I don't know, if you look for P300 in the ERP, still the latency or some features of the P300 could be affected by the previous trial or by a Q stimulus, etc. And so there is a toolbox called
See again. can come back.
Event-related potential by building a more informed design matrix. So in our case, for a task data, you would have the regular design matrix with the task, and then you could see whether find extra pseudo events with the blind approach, let's say, or in something in the middle, like naturalistic design, storytelling, etc. So you could also apply this and see whether, for example, the retrieved peaks correspond to particular salient events in the movie. So imagine that you have indeed a scan of, I don't know, 20 minutes watching a movie, and then in that case you could have, for example, a GLM, a blind GLM for, but not completely blind, let's say semi-blind, let's say like a semi-supervised learning, which you do it, for example, with all the frames which contain a face, or all the frames which contain movement, and then see whether there is a modulation of the retrieved HRF also in this case. Now I'm just hand waving, but let's say yes, it's applicable also to task data, depending on whether you want to be completely agnostic to the original design matrix, or you want to integrate this design matrix in this.
Do you
As
think,
a matter of fact, in one of the validation of our approach, we used some data recording from a rodent in which the somatosensory cortex was excited with some air puff. And yeah, in that case, we blinded ourselves to the design matrix and found the events triggering this. It was a quite easy case, if you ask me, but still, so you can use it if you want.
yeah, no, but that's good, because that's an engineering approach, right? You can't, if it doesn't work there, then you wonder about the higher level stuff. So that makes sense. And then if you're doing naturalistic stimuli, whether it's EEG or fMRI, I would assume that salience and attention are very closely linked, right? I mean, you know, so how, I don't know EEG particularly well. So how, on the neuronal side, do you see changes in, like, you know, P200s, whatever? Do you see attentional shifts if there are
I mean, also there, it's hugely important how you name things. Just today, I was in the guidance committee of a PhD student I co-supervised, and indeed his topic is about let's say perception and attention, and one of the members of the guidance committee say, okay, but can you even have, let's say, a prediction error without being attentive to it? So are the two things really impossible to disentangle? Yeah, of course, to some extent they are. So you always need to be careful of how you name things. So yeah, you can say, okay, maybe just, I mean, and this becomes more and more tangled when you go to more naturalistic settings, which basically which are the settings that you would like your research to live in, because then, of course, if you have the super basic task in which you can really have an attended stimulus and a not attended one, which are really stereotyped, etc. So, and maybe you can even do it, I don't know, while you are completely bored. As a matter of fact, some very basic stereotyped stimuli are used in passive brain-computer interface, even to probe your attention level. So if you perceive an oddball differently, it means that either you are in the flow while driving or while playing a video game, etc., or you are going to fall asleep, and then you need to epoch, let's say. So yeah, it's difficult to disentangle things in the same way in which is difficult to disentangle all the arousal and things. So for example, even in the last paper by Blaise Frederick's group, the one was just out in Nature Human Behavior or so, in which they say that the fluctuations correlate with arousal. Then, of course, I mean, I think it's no surprise at all for them and maybe for everyone, but it's difficult really to disentangle these two things, because I mean, among all the constructs that you can think of. Of course, arousal is the thing that one could name as the more related with the global physiology, let's say, not only neural activity, but also sympathetic and parasympathetic activity.
And it should be reflected in those measures too. So the heart rate should decline, the heart rate variability should decline. If you were measuring blood gases, or at least expired gases, you would expect to see changes there too. If you had pupillometry or some other measure, you would expect that to slowly, as they basically go towards sleep, right?
Yeah.
That makes perfect sense. That makes perfect sense. Yeah, I mean, at that level, I understand arousal. It's when it's when you're doing like we were talking about driving, and when you're in the flow state, whether you notice the woman about to step off the curb or whatever it is. That's really, really tricky because you're sort of on task. You're not necessarily on the task that is salient to the outcome, like getting A to B safely, but you might well be thinking very deep thoughts about Granger causality, right, while you're driving, right? And then you're on task. You're just on the wrong task for the so that becomes then attention is a thorny concept, or arousal is a thorny concept. You'd have to be very, very careful to make sure you're measuring the right things. Well, that's super cool. All right. So, do you want to try answering some of those questions that I wrote down?
Yeah, please. Yeah.
Okay, because like this is I listened to you guys, right? As I said, I watched your interview with Peter twice, and I sort of understood it a bit better the second time. And whenever I read a lot of these papers, most stuff goes over my head. For me, looking at this, I'm strictly on the acquisition side of this equation, and crap in, crap out. So what I need to know is how we can change things. I think this is again call to arms. What is it that we are doing wrongly currently that we should change, so that when you're trying to build models or tease out HRFs or whatever it is, maybe some lower hanging fruit rather than solving the brain, what should we change and why?
So, well, I think that, at least in my experience, from the acquisition side and the MRI engineering side or so, let's say it's really the part of science I most look up to, and from which I would really say, please never change, because it's really people that are really completely open to this in the practical side, but also knowledgeable about many aspects, and also in several cases agnostic to the old interpretation in terms of I don't know what will happen to the brain, physiology, etc. So, for example, one of let's say the most enriching experience for me when a few years ago I went in a sabbatical in a neurorehabilitation hospital in Venice was the interaction with an MRI engineer there, who basically, I mean, he had expertise in cardiac MRI, and then the same mindset. Then he had also collaborated in the developing human connectome acquisition, etc. So let's say he could really make use of all these things together. And so, for example, we came out with an idea of using the phase velocity scanner to scan the carotids and also the circular wheels. So to have a phase velocity curve by using a retrospective sequence triggered with a plethysmograph. So basically, if you want a resolution of 40 points in the cardiac cycle, you basically sample 40 cardiac cycles at different times, let's say. So basically something that in less than 1 minute you can do it. Indeed, we added our sequence and it was not, let's say, a very intensive things to do. And then you have a phase velocity curve at the carotids, and is remarkably stable across individuals and also different across individuals. So one of the things we are doing now
is
a
single measurement, right? This is not a time.
Yeah, it's a single measure, it's not a dynamic one.
So blood pressure. Okay, got it.
So once you have this, we were trying to do two things. First one is to put it in approaches such as the retro-ICOR. Instead of taking the PPG shape, just taking the onset of each cardiac cycle and then plugging in this phase velocity curve, simply because it's closer to the brain, if you want. And then to do an analysis similar to what we do with the HRF. So basically you have some shape parameters of this phase velocity curve, such as the height, the full width half maximum, the time to peak, etc. And to use them to see whether some aspects of functional connectivity, such as, for example, the correlation between the precuneus and the I don't know, and the frontal area, let's say a typical resting state pattern that you observe, depends also on this other type of transfer function. So let's say for me, from the acquisition part, I would say people who analyze data, I mean, like me, I mean, I'm not a psychologist myself. But then psychologists, neuroscientists, and so should basically, be more attentive to listen to people acquiring data. So I think basically you are really doing the right thing. I mean, you meaning people from the acquisition side. Then it's up to us to come with proper way to, I mean, to ask the correct question. Ultimately, it's all about asking the correct question. I mean, even something, let's say, a measure as imperfect as correlation. So everyone say, well, correlation is not causation, but I think that the people who invented correlation was not making fool of themselves. Okay, now this is actually a way to fool people to say that is causation or so. But on the other hand, they knew that it's just a measure of association. But if you apply it to two data sets, you are assuming that there is a relationship with these data sets. So you have a causal question that you want to answer. Then one say, okay, but then don't use correlation because it's a stupid measure. Then we can discuss about this. But on the other hand, in a properly controlled experiment and with a proper mindset, even correlation should be informative on causal questions. Then it's in the same way, even a tool which has all the limitation, if you want, and all the confounders of MRI and fMRI can, with a proper question, you don't need to build a new machine or a whole new protocol to answer meaningful questions. But you need good faith and you need the knowledge of the limitations, let's say.
Okay, all right, that makes sense. So yes, so you had the the implicit causal question in correlation. I understand that. I'm not a statistician, but I understand that, as you say, that there is you presuppose there's a relationship. Otherwise, you're looking for overlap. Otherwise, it doesn't make any sense. And that's something that's probably just, I'm sure people know this, they just don't think about it. It's not top of mind. So given that you're able to pull out HRFs from your resting state, and given that the standard way to do correlation, as far as I understand, is to take a single HRF, whether it's a gamma variant or whatever the flavor is, and then use that brain wide. So assuming that the HRF is the same brain wide. Does it?
No. It's one per voxel.
Okay.
So
okay. So you could take so you could take this from a resting state, then you could apply that voxel-wise HRF estimate, and you could apply that to your correlation in your task experiment. No?
Yeah. So there are two things you can do. Either if you want to do correlation between, for example, a voxel or average or PCA of voxels in region A and correlated with voxel in region B, then what I would advise to do is to apply our tool to deconvolve the HRF, and then to do the correlation between the deconvolved time series. This is one approach. The other approach is, on the other hand, to estimate the HRF in region A and the HRF in region B, and see whether the shape parameter of the HRF of region A and region B at the population level or the subject level in multiple scans, etc., so are associated with the same behavioral or pathophysiological condition that you are investigating.
Okay. Oh, okay. Yeah, that's a nice inversion. So you're basically
and then there is a third one, because I'm also in computational modeling and in generative models. So far you model brain activity either with some really basic models, or more biologically inspired model of neuronal activity with excitation, inhibition, GABA, glutamate, potassium channels, opening, closing, etc. So, and then you use a model for the hemodynamic response function. Typically is the balloon-Windkessel model, etc. So an alternative approach, if you have to fit some data, because typically you use this model to fit some data, is to estimate the resting state HRF from the data, and then to upsample this to your neuronal activity and to your synaptic activity, and then plug this into your model instead of the balloon-Windkessel model. So you would have a subject and region specific HRF already, but then depends again on what you want to model, because in this case, if you want to make inference about neural activity without being confounded by the transfer function, which is the HRF, then I would say do this. But then if you say, okay, I think there is something in the chemistry of the neurotransmitters and of the fact that of the neurovascular coupling, which will change this, then I think you will have to modulate also, to model also this, if you want to use a modeling approach. Otherwise, use an observational approach, and then this is what you have.
That makes sense. Yeah, then you're assuming a lot less about the system. That's probably a fair thing to do. All right. Okay. Then so going back to so just resting, especially resting state, because that's fraught, right? Because you don't really know anything at the moment about the system. You're just assuming that it's doing something and you can measure it.
Yeah.
What's your feeling on it then currently, given what you're experiencing with the modeling side? How confounded do you think most people's data really are? Do they even realize it?
Well, I think there are a different level of confusion and different levels of good versus bad faith, etc. So I think that a resting state per se is confounded. I would not say is much more confounded than the rest of fMRI, let's say, in terms of coupling, etc. So then, of course, if you have just a finger tapping task, then of course it's less confounded than a resting state. If you want to focus on only one region, I mean, you do a massive univariate approach, and that's it also. But let's say,
what about systemic effects, where finger tapping, you know, something, whereas
I mean, the systemic effects are definitely a big part of it. Now there was, for example, I mean, that paper by Blaise and Jun de Tong, in which they showed that even if you take a mock bold signal and then you just delay it in terms of the blood arrival time in different regions of the brain, you have something which resembles in terms of ICA, the different intrinsic connectivity networks. Or I saw this as a modeling person. I also saw that the fit to empirical fMRI data, if you use a biologically inspired model, or if you use just some mock bold or the PPG signal delayed by the blood arrival time, the fit is the same. Now, on one hand, you can interpret it as a tragedy, because you say, oh, you don't need neuronal activity. The human vascular system and also the systemic fluctuations arguably subserve neural activity, and vice versa. So the two things are not disconnected. It's not that you have something which is completely unrelated to neural activity, which recapitulates neural activity. Like, I mean, you cannot have neural activity alone, and you would not have that type of vascularization or that type of fluctuations in the brain if you didn't have neurons there, which needed to be perfused by blood. So let's say, arguably, physiology and maybe behavior in general are better measures, especially when it comes to clinics. You can treat probably better a person only by looking at their motor patterns or other vital indicators, and not exactly the neuronal fluctuation with fMRI. But on the other hand, let's say there is also an upside in considering this holistically. So yeah, I mean, not being cynical and saying the fMRI is just a very expensive, I don't know, motion detector, because actually it's motion which makes the difference between, I don't know, autistic people and people normally developing whatever. I mean, so and in the same way that you could just use, I don't know, a PPG and learn everything about the brain. But the fact that you say, okay, we can learn more things. And I think the time is ripe to use such a holistic approach without being afraid of this. Not even without making fools ourselves, because sometimes we become recursive, like all the global signal regression debate or so. I mean, there was in the last decade all sort of people, high-profile people, titling their paper, there is a signal in the noise, the rubbish of the brain contains information about the brain. But I mean, the global signal is literally the signal from all the brain. So you cannot be surprised that it's related to connection. Sorry, to cognition. It's just the biggest region of interest you can imagine. I mean, yeah, you could use the whole body if you want. If you could use
so it's single voxel fMRI.
But so it's definitely something that, if you apply, I don't know, global signal regression as if it was done in the end of the 90s, of course, you will induce negative correlation because of the difference in blood arrival time and of the fact that the global signal is differentially expressed in different regions of the brain, because some regions are contain more signal, some regions less, etc. So, but they're not two separate things.
Yeah.
In the same way that now, I don't know if you follow the new trends now in electrophysiology and EEG or so, with the aperiodic part of the spectrum. So let's say you have the spectrum with the alpha, beta peak, etc. And then of course, an alpha peak can change position either by itself or because the slope of the spectrum changes. And this is called the aperiodic part. But on the other hand, the aperiodic part and the periodic part of the spectrum are basically two faces of the same coin, the same way that the alpha and the beta, in our head, we separate them, but the brain doesn't care about this.
Yeah.
So even if you have a pure alpha oscillation, which suddenly turns into a pure beta oscillations, then, and if you take the spectrum over the we are agnostic to this. And so also there, the aperiodic part has been called the dark matter of the brain signal, etc. So it's not. But we should treasure these things and not, let's say, let's say, rain on our own parade or on our peers' works. Yeah, you see everything you see with the brain, you're just making a fool of yourself because it's just vascularization. I mean, it's just vascularization, but it's yet another proxy if you want of neural activity. It's
yeah. I mean, on the issue of information and your expensive motion detector, I've heard Gail Verquois make exactly that point very strongly. If you're chasing a biomarker, if that's your question, you're chasing a biomarker, then you become agnostic on whether it's if motion is the thing that carries the most signal, most information, then you go with that. And I think that's a strong point. And that then, if you go back to resting state fMRI, the default mode network, you could get into fist fights about people saying that, it's the default mode network a neural network or a vascular network. And I think that's to miss the point. It's a neurovascular network. They co-evolve.
Yeah, it is, exactly.
Right? They
as such,
exist in isolation.
It's basically sensitive to everything and specific to very little things. So you will find modulations of default mode networks in virtually all conditions and all tasks, let's say.
Yeah, well, of course. But also, if we didn't find that, the entire metabolic assumption under fMRI wouldn't hold. The
Yeah.
more neurons that are more highly active will require, on the average, a higher blood flow, which means they will be better vascularized. Ta-da! Right, shocker. Right, that's why layer 4 has all the, the granular layer has all
Yeah,
this
no,
stuff.
exactly.
Right, and so it becomes circular very quickly. So to ask, is it one thing or the other? It would be, I don't even know how, I can't even think of a good parallel for that outside of fMRI. But in a way, it's a specious argument, because you cannot separate these two. They are clearly neurovascular networks, and I think now if you want to, if your only interest is to make some statement about what neurons are doing, and you want to treat the blood as just some major domo that you'd prefer to that wasn't there, you've got a conceptual problem.
Yeah, I mean, and not even specific to fMRI, because I'm pretty sure that even not only at the very base of the signal, so all the f-haptic coupling, etc., so could be influenced by this. So it's not, I mean, even if you use EEG, but even if you use intracellular recordings. So I mean, I did 3 years of postdoc in a rodent and a cat electrophysiology. We were doing dynamic clamp in vivo, etc. So, and we were observing a lot of fluctuation with the heartbeat. It's not to say, oh no, there we were doing pure electrophysiology. There was a pipette with an electrode which stick literally into a single neuron. So
you mean there might be a
heart is not
sympathetic
important.
network where the neurons and the heart rate might be coupled? Are you kidding me? That's that outlandish statement.
Yeah. I mean, we saw glial cells because I mean the idea that you do in dynamic clamp is that once you patch a neuron, then you inject some current, different levels of increasing current, and then the neuron starts firing more and more. So you have the synaptic gain curve, which tells you something about then the function of the neuron. Then, of course, since also there we were going blindly into the brain of the animals, sometimes we patched a neuron and sometimes we patched a glial cell. Then in that case, well, you don't throw anything away. So you do the classical IV curve, current voltage curve also for the glial cell. And we saw that not only the voltage increased, but without spiking, because this was what you expected from a glial cell, but also that the resonance of the heartbeat in the membrane of the glial cell depended on the current that you injected into the glial cell, even if the glial cell doesn't spike. But so even in such a controlled system, you have literally one single cell you are monitoring, and you are injecting current in real time, because the dynamic clamp works like that. So you record the voltage from the cell, then you input this voltage in an equation, 40 kilohertz, and then you re-inject it into the neuron. And so you're basically mimicking opening and closing of ion channels, etc. So, and even there, even in such a perfectly controlled system, then you have the heart bit, which intervenes in respective ways. In the case you didn't have a respiration because the animals were paralyzed, I had a pneumothorax, etc. So they were still, but still you have this evidence for neurovascular coupling, even in such a tiny and perfectly controlled system.
And with massive amounts of feedback, where you've got
of course,
brainstem nuclei controlling the heart rate, the autonomic response, which then feeds forward into all of those neurons and glial cells, and then you're changing that local reactivity which you're measuring, and all you're doing is measuring what you get at the tip of your electrode, discounting all of this, that incredibly complex.
Also considering the fact that when we look at the human fMRI, it's not that the neurovascular coupling that you see happens inside the voxel. It happens at the level of the micro vessels rather than of the big vessels or vessels which are, let's say, of the size of a voxel, let's say.
And it might not be neural either. When we talk about neurovascular, we should probably say cellular vascular coupling, because there are smart astrocytes. We know that
sure. I mean, I was saying everything that generates the ball signal, let's say.
So, and there's an easy mechanism for variations in the HRF. It's just through arousal, through autonomic nervous responses, that as the brain prepares the animal for action or whatever it is, then those signals feed forward and modulate the astrocytes to the point where they start to modulate the blood flow. The neurons haven't changed yet, maybe, right?
Yeah.
It's quite possible that this is all non-neural and perfectly biologically plausible, evolutionarily advantageous. So yeah, there's many sides to this, and maybe that's another blind spot for us in a field, right? We keep citing neurons, and we should probably become cell biologists, not neurobiologists, right?
Yeah.
Maybe we're too neuro biased, and we treat the astrocytes or the glial cells as just another support system, and that may be causing us a major blind spot. That's a really interesting way to think about that. So okay, then for confounds, I spent a lot of time thinking about this, and I sent you that picture that I'm trying to write this review article on, and
Yeah.
it's just point every which way. Everything is circular because of the autonomic nervous system, and nothing is clean, right?
Yeah.
As I'm probably going to put in this paper, I hear people talking about throwing out the baby with the bathwater. You show me where the edges are, and I'll tell you if you can throw the baby out. I do not see edges. I just see blurry, right?
Yeah.
And also the blurring is not stationary.
Completely.
So I don't see how you can even think about baby and bathwater. They're all mixed up.
Yeah,
It's
no, indeed.
salad
Yeah.
dressing, right?
Yeah.
So when we think about confounds, or rather, maybe a better way to think about this is information that can shine light on what we're trying to interpret. Because I think here we're essentially talking about mechanism, and the default mode network is a good example, right? Where somebody wants to make a statement, a strong statement about neurons doing blah, and I come along and I say, yeah, but you might get the correct result, but your interpretation may be incorrect because it might be vascular, right? And again, this is sort of an argument. But when we talk about trying to understand the system, we are assuming that the fMRI is carrying the bulk of the information, and then everything else is somewhat peripheral, and we do some pulse ox, maybe some chest motion. And as I asked Dan Hamwork, I was just asking the same thing. What else should we be measuring, and why? What are we missing by doing such limited measurements? Should we be measuring arousal? Should we be doing pupillometry religiously?
Yeah, I mean, the evidence with pupillometry, I think, is quite clear. I mean, pupillometry has been shown to be a good function. A good parameter to correlate fMRI data and also EEG data in many cases. I would say again, in many cases in which you expect a certain level of arousal, and I would say in an unsurprising way. But then, of course, it doesn't have to be everything surprise. So we don't have to be surprised of things being coupled together, but also we don't have to be afraid of measuring things which in principle could tell us the same thing, because then even two things which look very similar one to each other, then with somehow with the proper modeling or analysis tools, so you can try to disambiguate different informational content coming from this. Now, for someone like me, let's say coming from, let's say, when I thought about interacting systems, when you think about a network where the nodes are completely separated. So let's say, okay, there is a cost associated, for example, to a link between, I don't know, New York and San Francisco in terms of airplane, because it costs fuel. So if you look at the network of connections of the road system in the Midwest where everything is flat, then it's a Gaussian thing. If you then look at airplane connections, then it's a power law connection. So we have some hubs, etc. So because there you have a much higher cost. In the same way, there is a metabolic cost in physical connection, etc. And even in some connections which are not tangible, like friendship, you cannot befriend everyone or so. So in this case, it's clear that the parts are separated. But in the brain or in the body, you cannot say, okay, I have one node, which is the insula, one node, which is the dorsolateral prefrontal cortex, and then I want to be open because I'm not only about the brain, and then one node is the heart, and then one node is the lungs, and one node is the liver, and one node is the pupil, but also there. It's not because they are in different anatomy textbooks. It means that they are separate nodes. They are all together, and so it's so maybe the paradigm that we should use is so even if in your scheme that I found beautiful and informative, even if it's messy, but we don't have to be afraid of messy things. The links are pairwise. So even if there are multiple links, you have links between I don't know, a perfusion pressure and CBF. There is a link with an arrow. Okay, sometimes I won't enter there because it really is not my field, but let's say sometimes we should just build those links, maybe not in a pairwise fashion. Maybe it should be some strings that we attach. So imagine that you, instead of these nodes, you have some pins. Instead of having a piece of string that you have also these memes of people connecting strings like crazy, etc. You can have a rubber band encompassing 3 or more parts or so, and this could be a functional hyperlink rather than several pairwise links. And then you can have this on the anatomical level, and then this on the statistical dependence level, and then you have a third level, at least a third one, which is the level of inference. So you have the level of biology, which usually you cannot observe in full. Then you have a level of statistical dependencies with all the confounders, etc. And then you have the level of inference of what you make of all this and how you interpret the results. So coming back to your original question, I would say let's not be afraid of measuring many things, and let's not be afraid of the obvious that some of those things will undoubtedly be redundant, one with respect to another. But in some redundancy, you could have some synergy hidden between it. And so in the trade-off between the synergy and redundancy or so, like in some experiments you do with the microgravity, then we live in a world with gravity, but still you can learn something by observing some things, and then knowing that then when you go to the real world, you will not observe them anymore. But
yeah, so right, so confounds have confounds, right? It's confound all the way down. And
also because it's difficult to call one thing a confound with respect to what, let's say, because my confounder could be your signal, and also even if all the mankind would disappear, just leaving the mechanism there free, then what is the confounder for a certain mechanism would be, on the other hand, the primary actor for another one. So it's
so right, and going back and thinking about arousal, you brought up the driving example, and again, just if all you have is a measure of arousal, that doesn't mean that you're necessarily on task. If the task is to drive and you're doing a driving task in the fMRI, and it turns out that when Daniele Marinazzo gets in the scanner, he's thinking about Granger causality intensely and barely paying any attention to the driving because you're that good. Exactly. That's confounded. We wouldn't know what you were thinking unless you told us, right? So you're on a task, just not the task. So that's a super important point. But so what I'm hearing then is we should measure more things, whatever that means. That more information, better. Recognize that everything is limited, but if we can at least get at a richer data set, maybe an EEG.
And also abandon the assumption that each of the items that we measure are actually separate items. So there could be part of the same system. We are just observing them in this way.
Yeah, absolutely. So that holistic approach, I think, is key, right? We are studying a biological organism, and then ultimately, I think that's why behavior is so important in all of this.
Yeah.
Trying to get at what was the organism doing? Were you paying attention to driving, or were you thinking about causality, right? And if you don't get that central part, then everything else starts to become really, really problematic. It would be like in a physical task, if it was just the person being active and you didn't know whether they were playing tennis or soccer, well, there's a big difference. But you know, but if all you were measuring was leg muscle activity, you might not be able to tell, right?
Yeah, no, completely. And then if I mean, tennis and soccer could be completely different things in one setting, but then if you compare tennis and soccer and just being a stone, then of course the tennis and soccer become much more similar one to each other.
Yeah, right. They are more similar than laying on the couch. Yeah, of course. Yeah. So again, behavior is key. Right, that's cool. So yes, so trying to capture more behavior maybe is even perhaps a better thing to do, especially with resting state. God knows how we did that, but that's just good food for thought. All right. Cool. We've been on an hour-ish. What else do you want to talk about? Colliders, I don't really understand. I think that to me, I think you've probably already answered this, which is there's no such thing as a confounder. Everything is, mixed up. So perhaps these are statistical constructs, or this is epistemological. It comes from people's training. It's not a reality when you get into biology, because everything is mixed up. Does that sort of make sense?
Yeah, definitely. I mean, and also the statistics that we do should be, let's say, tailored to the problems that we want to solve. So let's say there were two sort of illuminating articles I came across recently. So one was about someone who said, okay, people ask me which type of statistical should I use, for example, this statistical test or this other one or so versus this other one. And ultimately, the answer was basically the statistical test is always the same, is the null model versus another one. So you should build the correct null model, and this will automatically answer the question of which statistical test do I want to use. And then today there was just a blog post on statistics methodology blog called the 100% CI, as in confidence interval, which is called let's do statistics the other way around, which is indeed let's start from the problem we want to address, rather than say, okay, I have some observation, then I have this test which has conveniently been implemented in R, in SPS, whatever. So, and then I have the results, and then the results tells me this. Now, on the other hand, I say, okay, this is my hypothesis. This is why I did this experiment, and then I want to part of the experiment is to make sense of it with the statistics that stamps for it, let's say. So I would not say, okay, this is more a statistical rather than a methodological issue, or this is a statistical rather than acquisition issue. So ultimately, it's all about the question that you want to ask. So I think we should make some effort in wanting to ask the right questions, and then ultimately everything should come into pieces together.
Which, so what I hear there is that we have maybe a training issue, like we're becoming a bit too dogmatic, and we want to
definitely
break into the approach, because I think people
getting better, right? It's definitely getting better.
And this is where the incentives and just the structure of the way we do science gets in our own way, right? Because people come in grad school, then they want to graduate, they want to write a paper, all the usual stuff, and then they're in a hurry, always a bad sign, and then they are trying to get a result. The real goal, the question they're really asking is, can I get a job? Right? Not asking a scientific question per se, right? So now it's like, what statistical test should I use to get me over the line, right? Given that it's not then a scientific endeavor per se.
Yeah, I mean, even before, because sometimes, okay, the incentives sometimes are already within academia. So let's say a PhD student who wants to get a job or so, but even when we teach our students, so even students who don't even think about doing a PhD or whatever, but when we teach them, so for example, in my case, I started by teaching a course which was called Analysis of Neuroimaging Data. Then it's called now Technical Foundations of Techniques used in experimental and behavioral psychology and things like that. But the content is the same. It's just the title different to align a bit the expertise, etc. So it's selling the expectations. So saying that it's more about linear algebra and the signal processing stuff rather than the goal itself. So in the sense, it's let's say giving students the tool to understand some analysis tailored to this. But
so okay, so to use a cooking analogy, you're teaching people how to chop. You're teaching people how to saute, but you're not giving them a recipe, right? So
yeah, exactly. Or in the same way, because usually I see this in my evaluation scores, because sometimes I think they have some template in which things like the fil rouge, like how it's called, the red line, the common thread, and the big picture are mentioned because they are recurrent words in the evaluation that I received about student struggling to find the big picture and find the common thread. And I always say at the beginning, yeah, it's about understanding how a camera is made, so then you can have all the big pictures that you want.
yeah. That's a really important point. Yeah. So, and keeping that at least occasionally top of mind, so you don't lose that high level view, and just get into the as quantum physicists would put it, shut up and calculate, right? That you occasionally want us to think about spin and wave functions, right, and not just crank the machines to get the answers. That's cool. All right. Cool, man. This is fascinating. This went different directions than I thought it would. That's really cool. So anything else you wanna mention? Any other a new book coming out, or something to sell?
No, I mean, coming out, I mean, the things I already mentioned you about, this, putting this into a modeling in the computational model approach and so, and everything or so. But so for me, let's say the main topic was to answer your questions and what you find interesting or so. So it's not I don't have anything, let's say, that I want to make a statement here or to pitch anything. So
No, but I think you've given a lot of certainly I'm thinking differently already about, I thought, okay, I can just generate a list of extra things that we should measure, in or before or after the scan, and that will just help you a piece of cake, right? Basically, clean up the recipe, right? So, but it's what I'm hearing is that thinking more is what we really just need to do. Think about the
I think
question,
so.
think about levels, think about the organism, think about take a big step back. That's what I'm hearing. And we might still want to measure more things, but the answer to that question is it depends, because you have to start to the answer will differ depending on the question you're asking. If you're just trying to do an agnostic biomarker for disease X, it's one thing. If you're trying to understand a particular part of the brain for some reason, learning, whatever, then it's a different set of constraints. So that's cool. All right. Well, cool. So let me stop the recording. Thank you for this, mate.